We've seen a little bit about how powerful super-resolution techniques like STORM can be by allowing us to break the classical light microscopy diffraction limit and probe smaller spatial scales. But now let's look at how this can be used in practice to investigate biological systems and generate novel insights. Cellular structures such as organelles are usually large enough to be visible in optical microscopy, but not any of their fine details. Mitochondria are a good example. They're big enough to see clearly in a light microscope, but really just as dots. We can get an idea of their number and distribution, but only very rough morphological features. Biological structure often exists across several spatial scales, and in this case, if we wanted to study the mitochondrial associations with microtubules to form the mitochondrial networks, we would be unable to with light microscopy. This requires both a broad field of view to image the larger cellular architecture of the microtubules, but also very fine resolution to see subtle contacts between them. In this image, we can see the mitochondria with GFP, and there's some suggestion of a complex organization, but we can't really see the fine details. In order to overcome this, Huang used 3D storm localization to investigate the structure of the mitochondrial network in monkey cells. They imaged the mitochondrion outer membrane by staining TOM20, a translocase that's enriched there. This storm movie shows the reconstruction of the volume. And immediately, a few things are visible. In particular, the hollow space enclosed by the outer membrane. Interestingly, they were also able to distinguish cells with overall different mitochondrial morphologies. These probably represent different cell stages. On the right, there's a more elongated tubular network, which is probably associated with interphase. This network breaks apart during mitosis or apoptosis, producing the more globular, blobby mitochondrial seen on the left. They also used two-color staining to study mitochondria and microtubule interactions. Just using fluorescent microscopy, there isn't enough resolution to really see the sort of contacts there might be in any detail. STORM gives us more than enough resolution to see that there are actually mitochondria and microtubules in contact, which would be the case in transport, for example. They could also rule out some very close but false contacts that would be impossible with light microscopy. Additionally, there were some complex interactions, such as multiple or overlapping microtubule mitochondria contacts that we wouldn't be able to resolve with light microscopy. And so we needed this resolution in order to study the interactions of the tubular mitochondria with microtubules. They make pretty complex contacts, and they're not just lying flat on the microtubules. And so we actually even see some off-and-on type of contacts, making a sort of inchworm-like shape on the microtubules. And with light microscopy, again, this would just be a smear, and we'd lose any of the nice fine details we get with STORM. Another benefit of STORM is that it's possible to look at living cells, and we don't need to fix them. Other techniques like EM might require us to use harsh preparations that would kill the cell and remove any sort of dynamics. Shim demonstrated this, in fact, for a variety of cellular structures. So here's the plasma membrane, mitochondria, the ER, and lysosomes. In particular, the mitochondria STORM videos that they took showed potential intermediates to fission and fusion processes. You can see these as the thin tubular connections being formed and broken between neighbors. Remarkably, they were also able to see some transitions between the morphologies that we observed with Huang's work. This STORM video takes place over a minute, and in the course of it, we can see the thinner elongated network that's initially in place gradually becoming blobby and breaking up. This is probably a response to the photostress of imaging. We can also use two-color imaging in live cells to look at interactions between structures. Shim here studied mitochondria and ER connections. They were able to observe that ER tubules were often co-located with mitochondrial fusion, fission, or constriction sites. Of course, there's also a lot of other applications. Besides looking at organelles, we can look at individual biomolecules or complexes. So clathrin-coated pits were initially used as a test of the ability of STORM to accurately resolve biomolecular complexes. Here we have the ribosomes in a dividing bacterial cell. The cellular superstructure associated with particular complexes can also be revealed. Here, staining a particular calcium channel in sperm cells shows the entire flagellar morphology. This ability to span multiple spatial scales at the same time also leads to some of the most exciting applications of STORM in neuroscience. By labeling pre- and postsynaptic proteins and using 3D STORM, we can take accurate measurements of synaptic structural parameters. Since this is a relatively fast process, multiple synapses can be imaged and compiled into pretty large data sets. Then, using comparative analysis, we can reveal if there's any sort of heterogeneity or strong orientations or other measures of the synaptic geometry. In this work, Denny found that by clustering the various proteins, that certain scaffolding proteins were highly oriented in the presynapse, but also that there was a surprisingly wide lateral distribution of receptors in the postsynapse. If we move an architectural step higher, the organization of synapses in neurons can itself be studied. By imaging neuron volumes with GFP and locating synapses with STORM, Seagal showed that they have enough resolution, in fact, to accurately determine which particular synapses correspond to a particular neuron in a super-resolution volume. On the left is just the STORM volume, and the right is only the synapses that they found to be associated with the neuron. You can think of just trying this with a light microscope. This allows automated reconstruction of both the neuron's architecture and the associated synaptic architecture as well. This potentially revolutionary mapping of neuron architecture fundamentally relies on the particular advantages of STORM as a super-resolution technique by allowing the study of cellular organizations at widely separated spatial scales and then correlating that information. This particular advantage of STORM is going to be applied further by many groups in the future.